So this illustration by Rudolf Zallinger, known as the March of Progress, has attracted an army of criticism, most notably from Stephen Jay Gould, who opined that it misrepresents evolution as a predictable ladder of progress and not a copiously branching bush. The term superbugs, of course coined by the media, in a sense falls into the same trap. Superbugs are bacteria which have become adapted to specific environmental challenges, no different from any other adaptations in evolution. The traditional tree of life features Monera, the bacteria, as the primitive state at the bottom, and right at the pinnacle of evolution, in its German version, mentioned men. Of course, this is somewhat offensive, for nowadays we know that this tree of life should really be upside down with the bacteria at the top. And this is the reason why the amount of diversity in the bacteria is absolutely huge, compared, for example, with the tiny little group at the top, which encompasses mammals, birds and fish, which for some reason people tend to assume represents biodiversity. Now, superbugs is a term referring to antibiotic resistance. It's completely distinct from virulence, which is the ability to cause disease in the first place. Now, as I've got a bit of time on my hand, I'll talk about virulence first. So logic dictates that evolution should not favour virulence. If you're dependent on your host, then it makes no sense to harm your host. But if Mr. Spock had bothered studying for a PhD, he would have realised that there are circumstances when this trade-off doesn't exist. Cholera causes diarrhoea. The diarrhoea can get into the water supply. If you drink that water, you get more cholera, and thus virulence increases transmission. And John Snow famously showed how you can break this cycle by removing the handle from a water pump. Cholera contains genes which causes diarrhoea. The same genes are present in a very closely related bacteria which lives blamelessly in the beautiful bobtail squid. The squid doesn't get cholera, therefore these discased genes have nothing to do with disease. So the ability to cause disease is entirely context dependent, and we must always take into account that genes can move about from one species to another, often hitching a ride on these crazy lunar lander things which are viruses which infect different bacteria. Now, resistance. Penicillin was discovered by a Frenchman, Ernst Duchesne. It was rediscovered 30 years later by Alexander Fleming when he went on holiday and left his place to go mouldy. Florian Chain extracted penicillin just in time to save countless lives during the Second World War. Scary slide. Penicillin was first used in 1943. Resistance was first reported in 1946. Bacteria evolve quickly. There's lots of them and they replicate very, very fast. So MRSA was a national scandal in the 90s and the first half of the noughties, but you can see from this graph the rates tailed off quite dramatically, and that's due to hand-washing campaigns and hospital hygiene. It really isn't rocket science. So I'm interested in how MRSA spreads from one hospital to another. These red dots here represent MRSA from a hospital in Izmir, in eastern Turkey. The other coloured dots represent MRSA from Istanbul and Ankara. There's not much transmission between Izmir and those other cities. This is the UK. That's a copiously branching bush. These are hospitals in the UK. We've attempted to connect them on the basis of how much MRSA they share, how much transmission there is between these hospitals. We can look at the variation that's accrued during the spread of MRSA in the UK, and you can see there's a particular purple variant which is common to Ireland and some parts of Scotland. We also now know that the spread of MRSA in the UK originated in Derby, a particular event in the mid-1980s associated with the first use of an antibiotic called fluoroquinolones. The buzz picked up resistance to this antibiotic, which gave it a massive selective advantage, and it subsequently spread up and down the country. We can now also predict which antibiotics a particular strain is going to be resistant to, purely on the basis of the DNA sequence. We don't need to do any complicated experiments. This is going to put a lot of medical microbiologists out of a job. If you look at one particular region, in this case Portugal, you can see that at any point in time it's dominated by a single strain, the blue strain, then replaced by the yellow strain, then replaced by the red strain. This is a phenomenon called clonal replacement, and it's perhaps related to the fact that there is a cost to resistance, much like there is a cost to other evolutionary adaptations. In this case, this funny creature was thought to have gone extinct due to the fact that it grew these incredibly heavy armaments on its head and it couldn't lift its head up anymore. Kind of a weird idea. I also work on wildlife diseases of rainbow trout and honeybees, and vectors diseases, the body lice, and a zoonotic tick, which vectors Lyme. Thank you for your attention.